All right, so we have a really good summary slide of um, of the central dogma. It's slide number 12, and it says DNA and peptide formation. So at the very top of it, you have a double helix, and then we had said we have the genetic code. It's calling it the triplet seven. The triplet code. So the genetic code, sometimes called the triplet code. And we had said, what do? You make RNA that is complementary to the DNA. And so if you look at that number 2, it says this 7 base triplets on the template strand of DNA. And you see it's TAC, CGC, CCT, that blue structure across there. We had said you are going to produce messenger RNA, which is complementary to that. So A's and T's are complementary. And then when we're talking about RNA, not DNA, when you're talking about the complement to an A in RNA is a U. And then C's and G's are complementary. So if you look down below where it says transcription, the RNA that is produced by the triplet code, and we'll say the TAC, in that blue triplet code, it's AUG would be the complement to that. And then CGC would be GCG. This is the pink. Hopefully you're seeing it. Then the CCT is GGA, and so on. Well, the reason they started with that TAC, it wasn't by accident. It's because AUG is that start codon. So I'd said, when we're going to start translating that thing, that's where it's all going to begin. Okay, so we said that ribosome is moving down that sort of red-pink messenger RNA. We skipped over the alternative splicing at this point. But anyway, it's sliding along there, and as soon as it sees that AUG, it says, quick, bring the methionine. And so down below, we have the anticodons. And you see that AUG, the start codon, the anticodon that's on the transfer RNA that's carrying methionine, that MET, that's UAC. It comes in, and this is all happening sitting on the ribosome. And then the next codon has its anticodon on the transfer RNA, and it's bringing in alanine, and so on and so on. And so at the very bottom of this page, we have methionine, alanine, glycine, threonine, and so on. My glasses are giving me trouble. But anyway, the point is you build the protein that is coded for in the DNA. You see, the code determined that final polypeptide sequence. Okay, so we have that sequence of amino acids in a polypeptide. Now, what did we say when we were talking about ribosomes in the cell? We said sometimes they are translated, sometimes they're built into a protein on a free ribosome, and sometimes they go to that rough endoplasmic reticulum because they're going to be sent somewhere else. Well, it ends up at the very beginning of that messenger RNA, before you come to that AUG, there is what we call a signal peptide sequence. Now, what it does is it determines whether that protein that is coded in that messenger RNA is going to be translated on a free RNA or on the rough endoplasmic reticulum for secretion to the outside. So we have a signal peptide that is at the very beginning, and it's like a molecular address, and it has two choices: a free ribosome or rough endoplasmic reticulum. So it determines whether that protein stays in the cell or gets sent out and is secreted. Okay, now we still haven't gotten to the question that I mentioned a minute ago: the secondary, the tertiary, the quaternary structure. How do we get this folding? We said that these proteins, they only function if they're folded up. We said you can unfold them and ruin them, denature them. You can open them up and ruin them, make them non-functional. So how do we get them folded up? Well, it's something called a chaperone protein. Now, a chaperone protein, it's just like at a party, the older chaperone is there to watch over the youngsters, right? Well, the chaperone protein, as this polypeptide is being built, folds him up into the right shape. It's a protein it's another protein that combines with this forming protein to fold it up into the correct shape. That's what a chaperone protein is. Speaking of denatured, we had said there are various ways that proteins can be denatured. We had said, what were they? Temperature, right? And so we had said, well, obviously, when we're talking about chemical reactions in your body, you can't add heat. And so we had said we have enzymes that do this. But anyway, so I said that oftentimes people will die if you have high temperature, right? It's because it denatures proteins. But you don't always die when you've had fever, right? But what happens is some of these proteins get denatured. They get opened up where they don't function. So you have another kind of a big brother kind of protein. It's called a heat shock protein. And a heat shock protein does kind of the same thing as a chaperone, only it fixes the ones that have been damaged. Instead of being the big brother that folds you up in the beginning, chaperone protein, the heat shock protein comes in after you've gotten into trouble and got all bent up and puts you back into the right shape. And that is what their job is. And there are a whole lot of different heat shock proteins. There's one I'm just going to briefly mention. It's the heat shock 90. It's the cancer associated heat shock protein. Now, it's not one that is good. What it is there for is after cancer treatment. My wife worked in lung cancer research. And what happens is in lung cancer, the prognosis is not very good, even though it looks good at first. And it ends up that there's this heat shock 90. And so after you've killed back and gotten rid of most of those cancer cells, those lung cancer cells, this heat shock 90 comes in and fixes them. And so then oftentimes, tragically, lung cancers will spread. But it's because of this protein. And there was her most recent research had been involved with a drug that targeted heat shock protein 90. And it had huge promise. It looked like it was going to be a way to really help out lung cancer. But unfortunately, the drug worked beautifully. But when you first start making these drugs, you make them in tiny quantities, and you start testing them on mice where you've produced lung cancer, and they were living beautifully and it wasn't hurting the mouse. But then when you start making bigger and bigger quantities, it's harder to really make it exactly the same as when you're making it in a beaker. And it ended up during clinical trials, some people died. They died as a result of the treatment. And so that was the end of it. But if you can target this heat shock protein 90, it would be a really good one for helping to prevent this spreading metastasis of lung cancer and of cancers in general. And her work was with lung cancer. Let's see what else we've got on here. Okay, we've been 10 minutes. I have just a couple of more slides and we'll be done. So I'll be right back.